video. This video is about antigen processing and presentation to T lymphocytes, antigen presenting cells, and role of CD1 in antigen presentation. And this particular study note is developed by EPG Pathshala team, MHID. So I would like to express my sincere gratitude to the development team of this particular study note. Thank you so much. Our learning objectives include antigen processing is essential for T cell activation, self-MHC restriction, endogenous or cytosolic pathway, exogenous or endocytic pathway, antigen presenting cells, antigen presentation by MHC class II molecules, cross presentation of exogenous antigens, role of CD1 in antigen presentation. So after studying this module, you shall be able to differentiate between the modes of antigen presentation by B and T lymphocytes, understand the concept of self-MHC restriction, appreciate the need for antigen presentation to T lymphocytes, understand how the exogenous antigen are processed and presented, describe the processing and presentation of endogenous antigens, understand the role of antigen presenting cells, appreciate the concept of cross presentation, then understand how non-peptide antigens are processed and presented. So B and T lymphocytes differ greatly from each other in the approaches that they use for antigen recognition. The B cell receptor BCR, B and T lymphocytes differ greatly from each other in the approaches that they use for antigen recognition. B and T lymphocytes differ greatly from each other in the approaches that they use for antigen recognition. The B cell receptor BCR can recognize a specific antigen in its natural 3D conformation. Further, antigen recognition by BCR occurs directly, irrespective of whether the antigen is present on the surface of another cell or is drifting independently in the bloodstream or intestinal space within the tissue. Once again, let's learn this. B and T lymphocytes differ greatly from each other in the approaches that they use for antigen recognition. The B cell receptor can recognize a specific antigen in its natural 3D conformation. Further, antigen recognition by BCR occurs directly, irrespective of whether the antigen is present on the surface of another cell or is drifting independently in the bloodstream or interstitial spaces within the tissues. However, antigen recognition by the T cell receptor is an altogether different matter. A TCR can only recognize its specific antigen after it has been processed, digested into small peptides by another cell. Subsequently, the processed antigen peptide must be presented on the surface of this other cell in the context of a self-MHC molecule. Thus, TCR does not recognize a specific antigen in its natural 3D conformation but as small peptides. Further, antigens drifting independently in the bloodstream or interstitial tissue spaces are never directly recognized by TCRs. Such antigens are only recognized by the TCR after being processed and presented by an appropriate cell. Antigen processing is essential for T cell activation. Ziegler and Unane in 1981 and Shimokiewicz in 1983 provided conclusive proof that processing of the antigen into small peptides is a prerequisite for antigen recognition by the Th lymphocytes. These experiments showed that when an antigen presenting cell or APC presents an antigen to a Th cell, the Th cell with the cognate TCR gets activated. However, if the APC is fixed, killed by chemical treatment, and then an antigen is provided, the Th cell with the cognate TCR does not get activated. If, on the other hand, the APC is provided antigen 1 hour before it is fixed, then the APC is still capable of activating the Th cell with the cognate TCR. Surprisingly, even a fixed APC can activate the Th cell if the APC is provided with an antigen which has been pre-digested into peptides. All these observations prove that a Th lymphocyte can recognize an antigen only after it has been processed into smaller peptides. Studies by Townsend proved that activation of TC lymphocytes is also dependent on antigen processing. So, antigen presenting cells or APCs, antigen fixation antigen activates T cells. Fixation antigen does not activate T cell. Antigen fixation activates T cell. Fixation peptides activates T cell. So, flowchart depicting the experiments which proved that antigen processing is essential for T cell activation. Then self-MHC restriction. TCR of an organism can only recognize an antigen if it is bound to an MHC molecule belonging to its own MHC haplotype. This phenomenon is known as self-MHC restriction. TCR of an organism can only recognize an antigen if it is bound to an MHC molecule belonging to its own MHC haplotype. This phenomenon is known as self-MHC restriction. TCR of an organism can only recognize an antigen if it is bound to an MHC molecule belonging to its own MHC haplotype. This phenomenon is known as self-MHC restriction. Like MHC molecule belonging to its own MHC haplotype. So, Rosenthal and Shivas in 1973 provided experimental proof for self-MHC restriction of Th cells. Then peritoneal macrophages were isolated from guinea pig and were then incubated with the antigen. Meanwhile, the same antigen was introduced into guinea pig 2, and after a week, antigen primed T cells were isolated. These antigen primed T cells were then mixed with macrophages from guinea pig 1. So, guinea pig 1, isolation of macrophages, incubation of macrophage with antigen. Guinea pig 1, isolation of macrophages, then incubation of macrophage with antigen, then guinea pig 2, ingestion with antigen, isolation of antigen primed T cells, measurement of tritiated thymidine incorporation. Strategy used by scientists to prove that Th cells exhibit self-MHC restriction. T cell proliferation was estimated by quantifying DNA replication by measuring the incorporation of tritiated thymidine. The presence or absence of T cell proliferation depend on the haplotype of the two guinea pigs. T cell proliferation was observed only when the T cell and macrophage had the same haplotype, thereby demonstrating self-MHC restriction of Th cells. Strain of guinea pig, strain 2, strain 13, like that. Results this experiment. Guinea pig 1 provided the macrophage while guinea pig 2 provided the antigen primed T cells. Yes and no represent presence and absence of T cell proliferation. Scientists provided evidence for self-MHC restriction of TC cells. A CBAH strain mouse with H2K haplotype was immunized with the lymphocytic choriomeningitis virus (LCMV). After some days, its spleen was extracted and a splenocyte suspension was prepared. This splenocyte preparation contained TC cells specific for LCMV antigens. LCMV, immunization of mouse with LCMV, isolation of spleen after 7 days. Then spleen cells containing TC cells. Then fibroblast cell line, radio labeling with 51 CR incubation. So, meanwhile, a fibroblast cell line was either incubated with the LCMV virus or was left uninfected. Both these sets of fibroblasts were grown in culture medium containing radioactive isotype of chromium. Subsequently, both these 51 chromium labeled target cells uninfected with LCMV infected were incubated with the TC cells from CBAH strain mouse. The killing of the target cell was estimated by measuring the release of 51 CR into the culture supernatant chromium release assay. Only those target cells were killed which satisfied the following two conditions. They were infected with LCMV. Uninfected target cells were not killed. This observation suggested that antigen presentation was essential for cytotoxicity mediated by TC cells. They shared at least one MHC class 1 haplotype with the mouse, which was the source of the TC cell. This observation proved that TC cell exhibits self-MHC restriction. The experiment was repeated with mice having H2A and H2D haplotypes and even congenic mice acting as a source of TC cell. The inference from these experiments was also the same. TC cells exhibit self-MHC restriction. Then endogenous or cytosolic pathway. The pathway leading to the processing and presentation of endogenous proteins by MHC class 1 molecule is known as endogenous pathway. Endogenous proteins of the cell are broken down into two small peptides by a proteolytic complex, proteasome. Core proteasome is composed of 4 rings with 7 subunits each. The outer rings are composed of alpha subunits. The middle 2 rings are composed of beta subunits. Viral infections lead to an increase in interferon, IF and gamma levels, which result in the conversion of the proteasome into an immunoproteasome. In the immunoproteasome, the subunits beta 1 and beta 2 and beta 5 are replaced by beta 1I, beta 2I and beta 5I respectively. Conversion of proteasome to immunoproteasome leads to increased antigen presentation by MHC class 1 molecule. After the antigen is processed into small peptides in the cytosol, it is transported into RER. This transport is an ATP-dependent process mediated by TAP, transporter associated with antigen processing. TAP preferentially transports those peptides whose C termini are basic or hydrophobic. The TAP preferentially transports those peptides whose C termini are basic or hydrophobic. The TAP-associated protein, tapesin, binds to ERP57 and recruits the calreticulin MHC class 1 and ERP57 complex to the TAP. The multi-protein complex resulting from this association is known as peptide loading complex, PLC. Besides connecting the MHC class 1 molecule to TAP, tapesin also performs peptide editing, a process which involves exchange of low affinity peptides on MHC class 1 molecules with high affinity peptides. The ideal site of peptides for loading on class 1 MHC molecule is 8 to 9 amino acids. PLC loads the right sized peptides on MHC class 1 molecules straight away without exposing them to ER lumen. However, longer peptides are trimmed from their amino terminus by the enzyme ER aminopeptidase 1, ERAPI, P1. After the desired length is reached, PLC loads the peptide on the MHC class 2 molecule. The binding of the peptide to MHC class 1 molecule stabilizes it. The peptide MHC, PMHC complex then gets dissociated from the PLC. From RER, the PMHC complex moves to the Golgi apparatus and is then presented on the cell surface. In case of virally infected or altered cells, ASCs, such as cancerous or aging body cells, the peptide MHC complex may be recognized by a cognate TCR on a memory TC cell. In the presence of appropriate co-stimulatory signals, the memory TC cells will get activated and would then become a CTL, which would eventually eliminate the virally infected or altered self cells. However, activation of new T cells is usually brought about by a type of APC called the dendritic cell. Endogenous pathway. For convenience, the pathway has been shown in 12 steps. Step 1 and 2 show the assembly of MHC class 1 molecule wherein the heavy chain of MHC class 1 gets folded and bound to beta 2M with the help of calnexin and ERP57. The MHC class 1 molecule then interact with calreticulin. Wait a minute. Calreticulin and tapesin. So first, assembly of MHC class 1 molecule wherein the heavy chain of MHC class 1 gets folded and bound to beta 2M with the help of calnexin and ERP57. The MHC class 1 molecule then interact with calreticulin and tapesin. Tapesin links the MHC class 1 complex to TAP. Represent the translation and proteasome in mediated degradation of endogenous proteins. Step 7 depicts TAP mediated transport of peptidase from cytosol into RER. Step 8 illustrates ERAP1 mediated trimming of peptidase. Step 9 and 10 show PLC mediated transfer of peptidase to MHC class 1 molecule. The transport of peptidase MHC complex from RNA to Golgi apparatus is shown in step 11 and from there to plasma membrane in step 12. Exogenous or endocytic pathway.